Good afternoon, everyone. I'm Yuzhi Lin. It's my honor to talk about my research study in National Tsinghua University in Taiwan with my advisor, Dr. Zhonghong Lin. The topic I want to introduce today is using solid-liquid contact electrification for carbohydrate protein interaction studied, and is used for label-free bacterial detection. Bacteria is everywhere in our daily life. According to a report recently released by the World Health Organization, nearly 2 billion people worldwide use facial contaminated water sources, putting them at the risk of trachea, cholera, and dysentery. And every year, more than 500,000 people die from diarrhea caused by polluted water. 75% of bacteria that commonly cause diarrhea are urethral pathogenic Escherichia coli, referred to as UPEC, which is prone to intestinal and extra-intestinal infections, such as urine infections. Among the isolate of UPECs, more than 95% was found to have type 1 phyllid, which are easy to attach to the epithelial cells of human body, causing urinary tract infection, inflammation of bladder, or inflammatory bowel disease. Therefore, rapid, accurate, and specific detection of these bacteria in water has become a basic requirement for water quality assessment. However, current detection methods of biological samples such as PCR, fluorescent detection, or flow cytometry not only require complicated sample pre-processing, sample culturing takes 24 to 72 hours, also requires the use of costly and bulky instruments with professional techniques for detection. Hence, we propose a new detection method based on the principle of solid-liquid contact electrification by measuring the voltage output as our signal source. This detection method is not only label-free, rapid, simple, low-cost, and portable. There are several kinds of contact electrification. Here we propose to use solid and liquid contact electrification is because of liquid can make sure total contact and separation with solid surface and overcome the wear between the solid-solid surface. Furthermore, the triboelectric output generated from solid-liquid TENG is not influenced by environmental factors such as humidity. Based on this concept, we developed the first solid-liquid TENS applied in biosensors field. Our sensor design concept is first grow a layer of gold nanoparticle. From the SEM image, we can confirm that the uniform size of gold nanoparticle with 100 nanometer. And from the EDEX, we can confirm that our sensor is without impurity. And then we modify D-mannose on the gold nanoparticle surface by using FTIR to analyze, comparing after reaction with D-mannose. These three strong vibration bands confirm that D-mannose is successfully modified on our sensor. When the sensor grabs the target, the morphology and composition of the sensor surface will change, so the work function will be different. Therefore, in this study, the work function of the sample surface played an important role in determining the voltage output. The core mechanism of our solid-liquid TENS is that when the target is attached to the sample surface, the work function changes, resulting in a response to the difference in voltage output. By contact and separation the liquid and solid, there will be an alternative electron flow in the external circuit, becoming output voltage. Because of type 1 fillet is composed of protein, first we use a common protein which is cone A to verify the mechanism of our sensor. UPS analysis shows that after sensor is connected to cone A, the work function is gradually reduced from 6.72 electron volt to 5.32 electron volt. The right figure is schematic illustration of triboelectric charge transfer process during contact electrification between cone A and PBS. We also found that as the concentration of cone A increased, the output also increased with a positive trend. By using KPFM analysis, from the KPFM data, we can know that as the concentration of cone A increased, the surface potential also increased. The trend of KPFM and UPS match with the result of the increase in voltage output, verifying our core mechanism. We also did a reusability test. After the target was detected and determined, in order to destroy the specific binding between the target and D-mannose, our sensor was immersed in a high concentration of D-mannose solution. The attached cone A will tend to bind to the higher concentration of D-mannose, thereby removing from the sensor surface. KPFM was used to analyze the surface potential between each step. The surface potential after the reaction with a high concentration D-mannose solution dropped to the DNS surface potential before the reaction with cone A. This positive result directly verified the usability of our TENS. We verified the feasibility of this method by lactin, so we extended to the detection of bacteria. From this SEM image of bacteria with type 1 fillet, there are many fillet outside the bacteria surface, and there is fin H protein on the top of the type 1 fillet. From the reference, we know that fin H binds specifically to D-mannose. Therefore, we can easily use our sensor, which modified with D-mannose to catch bacteria with type 1 fillet. In this study, we select E. coli ORN178 as our target, and E. coli ORN208 and S. aureus as our control group. Similarly, in the ORN178 group, we observed the same increase in output voltage as concentration increased, but the same trend was not observed in other two groups. Through this experiment, it shows the great sensitivity and selectivity of TENS for detecting different concentrations of E. coli with type 1 fillet. The conclusion is that we have successfully demonstrated a self-powered sensor based on a solid-liquid contact electrification mechanism for detecting biological material. This method is not only fast, cheap, reusable, but also highly sensitive and selective. Such novel detection method shows great potential in the future to detect bacteria and even other biological samples, such as protein and DNA in actual samples. In the end, I'm appreciative to my Professor Zhonghong Lin, the help from my team members, and the support from NTHU, BME, and MOST. Thanks for your attention.